how the proteins are transferred from one part of the cell to other part of the cell. For example, how the vesicles transferred from endoplasmic reticulum to Golgi. Next, how the vesicles transferred from Golgi to the other part of the cell. Right.
Yes,
All these movements takes place due to the cytoskeleton. Got it?
Yes, ma'am.
Yeah. So these are the proteins. Okay, cytoskeletons nothing but proteins responsible for providing rigid structure to the cell, providing intracellular movement, and it helps for mechanical strength to the cells.
Mechanical strength.
Okay, mechanical strength or mechanical support to the cells. Understood? So for we have skeleton system, right? So that's why we have particular body shape. Am I right?
Absolutely, ma'am.
Similar to that, so some cells are spherical, some cells are cuboidal, right? And some cells are elongated, some cells are the cuboidal, or some cells are polyhedral, right? So different shapes are there in different type of cells. Who give the shape? The cytoskeleton. And in eukaryotes, as we already studied, in eukaryote, so nucleus is centrally placed, right? So nucleus is nearly spherical in shape. Okay. So this spherical structure of the nucleus is also due to the cytoskeleton.
Ma'am, so like I have a question. So nucleus being like two membrane organelle.
Yes. Nucleus is double membranous organelle. Okay. Inside the double membrane, so the cytoskeleton protein layer present, protein filament. Okay. So this is a thin transparent layer that gives shape and support to the nuclear membrane.
Okay, ma'am.
That is called nuclear lamina. I told you, no? Nuclear lamina. We studied this. Lamina, nuclear lamina. So it is made up of cytoskeleton proteins.
Yes, ma'am.
Clear here?
Yes, ma'am.
Yes. So let's start the topic, cytoskeletons. New student join us, Snigdha.
Yes, ma'am.
Are you joining for zoology class?
Yes, ma'am. I plan on doing microbiology and zoology.
So you are joining for GATE Life Science. Tell me your subjects.
I'm taking, of course, chemistry and microbiology.
Yes. Okay,
and
next.
Zoology.
Microbiology and one more is?
Zoology.
Zoology. Then why you didn't attend the previous classes?
For some reason, I wasn't added to the group, ma'am. I was only added very recently. So I had to talk with the GATE management to let them add me to the group.
Okay, few classes are completed, Snigdha. I started cell biology part, cell biology unit. Okay.
Okay, ma'am.
Yes. This is the first topic, cell biology unit. Okay, so in cell biology, two topics are completed. Cell membrane structure and cell organelles. Two parts completed.
Okay, ma'am.
Okay, Snigdha, do one thing. So in your portal, so previous classes are recorded.
Okay, ma'am.
Okay, please start from the first class. In first class, I explained how to start the preparation, 6 months plan, okay, course plan, monthly plan, weekly plan, day plan, how to start your everyday preparation. Everything I explained in the first class, introduction class.
Okay, ma'am.
Okay, please refer the first class. After referring that, if you want any other extra information or you want the different way of teaching or any other suggestion or any other issues, contact me directly.
Definitely, ma'am.
Okay, so we'll help you. So what you want to reach your goals. Okay.
Sure, ma'am.
Yeah. Okay. So this is the ongoing class. So please parallelly go through all these. Okay. Ongoing classes with previous classes. Okay. Parallelly refer all these. And within 15 days, please finish off all these previous classes.
Sure, ma'am. I will.
Okay. Okay, ma'am. Sure. After completing the membrane structure part, inform me. We'll conduct test for you.
Okay, ma'am. I'll let you
Okay.
know.
After conducting each topic, we'll conduct test.
Okay, ma'am.
Okay. You should inform me after reading revision of the topic.
Okay, so you have finished 2 topics, so I will need to take tests.
2 tests. Yes, you need to take 2 tests.
Okay, ma'am.
Okay, I conducted 1 test. So one more test is pending. Next week we'll conduct the test.
Okay,
Because
ma'am.
one more student, so is not she's not completed the topic reading revision. So that's why. So we are conducting testing next week. Yeah, it is fine.
Yes, ma'am. Okay.
Okay. Let's continue the next topic. Cytoskeleton and cell motility. And Snigdha, regarding the study materials, we'll share you separately. Okay, refer those materials.
Okay, ma'am.
Next topic, cytoskeleton and cell motility. So eukaryotic cells possess cytoskeletal system. Okay. So here the cytoskeletal system gives structural support to the cell. Okay. And cytoskeleton system composed of 3 well-defined filamentous structures. Okay. You know, there is a cell. Okay. Cell is cuboidal in nature, and this cell has many organelles. Okay. And these organelles are interlinked. Okay. And these organelles perform the coordinated functions. Right. And you know, these organelles are situated at the particular region in the cell. Their nucleus is present at the center. Next to the nucleus is endoplasmic reticulum. Next to that is Golgi apparatus. Right. So in particular position, the cell organelles are located. Okay. And the shape of the cell, support of the cell, and the position of the cell organelles is given by the cytoskeletal filamentous structures. Cytoskeletal filamentous structures. Okay. These cytoskeletal filamentous structures are 3 types. Mark this. This is very important for exam. Those 3 elements are, those 3 filaments are microtubules, microfilaments, and intermediate filaments. This is very important, students. Please remember, mark this. So each of the 3 cytoskeletal elements are made up of polymer of protein subunits. Okay. And here these protein subunits are joined by weak non-covalent bonds. Please remember here why weak interactions takes place in the proteins. Here many globular proteins are there, right? See, one protein subunit interact with the other one. See, like that many protein subunits are interacted by non-covalent bonds. Okay, so these non-covalent bonds help for rapid assembly of the proteins and rapid disassembly of the protein, like this. Rapid assembly and disassembly of the protein. Got it? So this assembly and disassembly of the proteins help for cellular regulation. Okay, cell functions regulations. So later topics will tell you with example what it means. Okay, so here just remember the statements. Okay, so let's discuss one by one. First one, microtubules. Look at the second image. This one. The microtubules are long, hollow, unbranched tubes composed of protein tubulin. See, this is one tubulin. This is another tubulin. See, many tubulin proteins are joined together to form the tube-like structure. Okay, see, tube-like filament, hollow tube-like filament. So this is the microtubule filament. Microtubule filament made up of tubulin protein. Clear everyone? See, their diameter is 25 nanometer.
Yes, ma'am.
Second one, microfilament. So microfilament is solid thinner structure. See, it is like a helical structure. See? Double helical structure. Not completely helical, it is like a helical structure. Okay, so these are made up of the long protein subunit chain is called actin. Okay, so many actin proteins are joined together to form the microfilament.
Microfilament.
The microfilament diameter is just 6 nanometer. It is very thin. Okay, thin and smaller one. Got it? Next, third one, intermediate filaments. The intermediate filaments have tough rope-like fibers. See, many fibers are there. Long thread-like fibers are there, right? So many fibers are parallel to each other. Many protein fibers arranged parallel to each other to form the intermediate filament. Here, not the same one type of protein. Many related proteins are joined together to form intermediate filament. For intermediate filament, there is no proper one protein. Many related proteins are involved to form or joined together to form intermediate filament. Got it?
Yes.
For microfilament, actin proteins are present. For microtubule, tubulin protein. For intermediate, many proteins involved. Clear?
Yes, ma'am. Clear.
Snigdha, understood?
Yes, ma'am.
So these are the three types of cytoskeletal filaments. Okay, read this slide once. Understood?
Yes, ma'am.
Okay. Functions of cytoskeleton. Cytoskeleton provides structural support that determines the shape of the cell, and it helps for positioning the various organelles within the cell, and it helps for intracellular movement. Okay. And it helps for independent movement of the cells. Because do you know flagella and cilia?
Yes, ma'am.
Do you know? Say these flagella and cilia.
Organelles.
All these are made up of what? Cytoskeletal filament.
Cytoskeleton. Yes, ma'am.
Okay, and you know, and the cytoskeletal filaments are essential for the cell division. Okay, so one cell undergo division to form two cells. Okay, so this division is also due to the cytoskeletal element. Do you know spindle fibers during cell division?
Yes, ma'am.
Yeah, those spindle fibers made up of cytoskeletal element, that is actin protein. Okay, so
yes,
these are the various important functions of cytoskeleton. Okay, look at the image. See, this is the organization of cytoskeletal elements in this cell. Refer this diagram carefully. Just a minute, student. We'll come back.
Yes, ma'am.
Yeah. Completed?
Yes, ma'am. Ma'am, so here for the microtubule, why is it showing like alpha tubulin and beta tubulin?
Here, repeat again. Your voice is breaking. I can't
Ma'am,
hear
I was
properly.
saying, can you hear me? Hello?
No, here it's breaking. There is a network issue, I think.
Okay, ma'am.
Can you hear me?
Ma'am, I can hear you.
I can hear you, but voice is breaking. There is no clarity.
Okay, ma'am. I'll just write my question.
Can you join again? Here.
Can you see my question, ma'am? Hello?
Did she leave the meeting by any chance? I can see your question and I can hear it clearly.
Oh, okay. Hello? Now can you hear me, ma'am? Hello?
I think she has some network issue and she just left the
Okay.
Zoom meeting.
Okay, I think she is joined back for participants.
Students, can you hear me now?
Yes, ma'am. We can hear you.
Yeah, here is too rainy, ma. So there is a network issue, server issue is there.
Okay, ma'am.
If you can't hear, please let me know.
No, ma'am. I could hear you
From morning
clearly.
only, network issues are there.
Okay, ma'am. Ma'am, so also my question was like, what is the difference between the monomers of protein tubulin? Like there was alpha and beta, no? Like so, how are they like different from each other?
And that will explain you.
Oh, okay, ma'am.
Structure and all will explain. Okay, so here just see so how the three different cytoskeletal elements are arranged in the cell. Okay, see here there is a mitochondria, right? See mitochondria is supported by this microtubules, right? See green color is the intermediate tubule, right?
I'm not able to see
The
your
intermediate
slide.
tubules are there and the blue color. This is the microtubule.
Hello, ma'am. I'm not able
Can't
to
see
see
this
your
thing?
slide. Now I can see it. Yes, ma'am.
Yeah. Okay. So here there is a mitochondria. So mitochondria is supported by, see green color is the intermediate filament. Okay. See intermediate filaments and see blue color one. Microtubules. Okay. So then here purple colors are tubules are there. See here,
Yes.
the location of the three elements are different in same cell. One cell, the location is not same. Right? It is different. Some are attached to the cell organelles, some are present towards the cell membrane, some are present towards the cell center, right? In different position, so different cell organelles are situated. And these are supported by the microtubules, microfilaments, and
intermediate filament.
the intermediate filaments. Yes. Alpha and beta tubulin will explain. Okay, let's start the first one, microtubules. Okay, so microtubules, as you know, the microtubules contains tubulin protein subunits, right? And microtubules acts as structural supports and organizer. Mark this. This is very important. Microtubule major function is it helps for structural support and it helps for intracellular movement. Protein vesicles are transferred from endoplasmic to Golgi. Intra means within cell. Okay, endoplasmic reticulum release proteins in the form of vesicle. Okay, so this protein vesicle transferred to the Golgi through these microtubules. Okay.
Yes, ma'am.
The next see, so the microtubules have the protein subunits and protein subunits are two types. Okay. Alpha tubulin and beta tubulin proteins. Alpha tubulin and beta tubulin. Two types of proteins. These are same, but only just these are the isomers. Only some structural changes are there. Okay. And first is alpha tubulin. Next is beta tubulin arranged joined. Next is alpha tubulin. Next is beta. Like that, the alternate tubulin subunits are joined. Okay, so one end, one terminal is alpha tubulin, other terminal contain beta tubulin. Okay, so the alpha tubulin end is called minus end. Okay, beta tubulin end is called plus end. Okay, plus end and minus end. So now we will get the idea. So one microtubule have plus end and minus end. Plus end means beta
positive
tubulin.
side.
Positive side. Beta tubulin subunit end. Okay. So minus means alpha tubulin subunit end. Got it?
Yes.
This is very important for functioning. Okay. Please remember this. And so these, for example, so we are traveling from house to the other place like college. Okay. So from house to college, there is a path, right? So there is a road, through road. So we are traveling from house to college, then college to house, right? Through
Yes.
road. Here, road is the
Hello.
okay. So road provides movement for the movement from house to the college, right? So without road, can we travel?
No, ma'am.
No, right? Similar to that. Okay. So and so from house to college, so we are traveling on road with the help of any vehicle, right? Or through walk.
Walking.
So through legs.
Ma'am, you're not clear.
Okay. So here road is the microtubule. Please remember, road is the microtubule and vehicle is the motor protein. Vehicle is the motor protein. Got it?
Yes, ma'am.
So on the microtubule, motor proteins are associated and motor protein transport vesicle from one part to other part. What is diagram? See here, this is the road. Okay, this is the microtubule. Okay, on the microtubule, see there are some motor proteins. These motor proteins helps to transfer vesicle from one part to other part. Okay, so here road is the microtubule, vehicle is the motor protein. Okay, let's discuss the motor proteins of the microtubule. So different cytoskeletal elements have different motor proteins. Okay, so motor proteins are important for transport of the vesicles. Okay, motor proteins have the ability to generate energy. Okay, so energy will get from ATP hydrolysis. I will tell you later. Okay, let's discuss motor proteins of the microtubule. First one, kinesin. Two motor proteins are there in microtubules. First one is kinesin. So kinesin is a tetramer. Kinesin is a tetramer consists of two identical heavy chains and two identical light chains. Here these two are the heavy chains. Okay. And these two are the light chains. So there are several parts. See, globular head, neck, okay, then stab, and here this is tail. Okay, so here this is the head. The head has the ATP generating engine. Okay, so it has the ATP binding site, and here ATP hydrolysis takes place.
Where, ma'am? Hello?
So in head, ATP hydrolysis takes place. You know, ATP hydrolysis gives energy to the motor protein, and now motor protein helps to transport vesicle from one part to other part.
Okay.
Okay. Okay. So look at this image. Here you will get the clear idea how the kinesin protein transfer vesicle. See, this is the head of the kinesin. This is star. That is type. Okay. So here head binds to the microtubule protein. That is tubular protein. Okay. So now it concludes it has two binding sites, right? One is ATP binding site, other one is microtubule binding site. Tubulin protein binding site. Head contain two binding sites. Got it? It is like a leg. Understood?
Yes, ma'am.
See, it binds to the microtubule, okay, and so it binds to the ATP. Now ATP gives energy for transport this vesicle. So tile binds the vesicle or transport particle, anything. Okay, so here ATP undergo hydrolysis to give energy for the motor protein. Now kinesin is activated and transport vesicle in this direction. Okay. And kinesin always undergo movement from minus to plus end. Minus to plus end of the microtubule.
Movement from.
Understood everyone?
Yes, ma'am. Minus 2 plus end.
The
So
microtubule is
it
like a
starts. So it starts from alpha tubulin end and ends at beta.
yes, correct.
Yes, ma'am.
Yeah.
Hmm.
So these are the alpha tubulin subunit, these are beta, these are alpha, these are beta, like that.
Hmm.
Okay.
Got it, ma'am.
So yeah, see these are like the legs. Okay, so these are moving in this direction. Okay, so we are walking means due to energy in our body. If there
Hmm.
is no energy in our body means there is no muscle movement. There is no muscle movement means we can't move. Same like that. So the kinesin molecules are also undergo movement with the help of energy. That
Hmm.
energy in the form of ATP. Clear everyone?
Clear, ma'am.
Yes, ma'am.
Okay. Next, dianin protein. It is another motor protein in microtubule. So dianin responsible for cilia and flagella movement. Okay.
Hmm.
So the complex molecule, okay, multi subunit complex of dianin protein is called dynactin. Okay, so dynactin has a globular head and many intermediate light chains are present. Okay. So dynein transport substances opposite to the kinesin. Dynein transport substances towards the minus end.
So
Towards
it's
minus end.
just the opposite. It starts from
Ah,
beta tubulin end and it ends at alpha.
yes, opposite to the kinesin.
Ma'am, so like I
Okay.
had a question. Do these work like simultaneously or do they
It
have
can,
different?
it can. It can.
Okay.
There is no relation for these two. Okay, they perform their function simultaneously. Okay, or whenever transportation requires. So these are ready. Okay,
Yes,
but
ma'am.
these are requires energy for the transportation.
Mm-hmm.
Look at the image. Here Golgi complex is there. So Golgi complex release vesicle. So this vesicle transfers from Golgi to the plasma membrane.
Hmm.
Okay. So here microtubule present here. So minus end is towards Golgi, plus end is towards plasma membrane. So now which one is transport vesicle from Golgi to plasma? Kinesin, right? Kinesin
Kindness.
transport vesicle from minus end of the microtubule to plus end.
Hmm.
Okay. For example, plasma membrane have the lysosome. It need to transfer towards the Golgi. Who helps to transport? Dynein. Because dynein transports substances from plus to minus.
Plus to minus. Hmm.
Okay, it can take place simultaneously. Okay, but in a different site,
Hmm.
not on the same site. Okay, different tubule. Clear everyone?
Yeah.
Yes, ma'am.
Yeah. See, so many other proteins are also involved in the movement of the vesicle. For example, this is vesicle. See, vesicle have the membrane protein. Okay, so membrane protein binds to ankyrin protein, then spectrin protein, then the dynein protein, then microtubule. So all these complex structure is called dynactin. See, spectrin ankyrin is also a cytoskeletal protein. It's not a major, but it's a cytoskeletal protein. Got it? See, for transport of one vesicle, all these are integrated and binds each other and perform their action. Okay, so this is the mechanism of intracellular movement. Got it? So the ankyrin and spectrin are the accessory proteins. Accessory proteins. Remember. Look at the image. So if minus with means microtubule usually have minus end at the center, plus end at the periphery.
Plasma side. Plasma
Yes.
membrane side.
Yes, peripheral side.
Ma'am, so is this in the center is this green Easter egg kind of thing the nucleus? Because I can't see. What is that? That center में green thing?
Centrosome. Centrosome.
Oh, centrosome. Okay.
Yes. Because centrosome have the ability to synthesize the cytoskeletal elements. So at the center, see two perpendicularly arranged structures are given, right? These two are the centrioles. These two are centrioles. Got it?
Yes, ma'am.
Yes. So kinesin transports substances towards the cell periphery, means towards plus end of the microtubule. Dynein transports substances towards minus end of the microtubule, means cell center.
So, ma'am, is that the nucleus?
No, it is a center, nothing but anything. So, near to the nucleus, centrosome present.
Okay.
For understanding purpose, they given like this.
Okay. Okay.
Yes. Next. So these microtubules functions and motor protein functions are regulated by some chemicals. Like say colchicine, it is an alkaloid. So it inhibits the assembly of the tubulin subunits. Okay, so tubulin subunits are need to join like this, right? But this assembly of the tubulin subunits is inhibited by this alkaloid. Okay, and so this is the drug. Nocardazole is the drug. This also inhibit the microtubule assembly. Okay. And vinblastine, vincristine, these are the alkaloids. These are also cause aggregation of the tubulins, suppression. Okay. So
ma'am,
like
what is this autumn crocus?
That is a plant.
Okay.
One type of plant. From that, these alkaloids are produced. Got it? So next, Taxol, it is also a one drug, anti-cancer drug. Okay, so because Taxol inhibit the cell division, because cell division takes place by the help of spindle fibers, but spindle fibers is not produced because of this drugs, because this drug inhibit the formation of spindle fiber that inhibit the cell division. So cancer condition. So one cell is tumor, means neighboring cells are also becomes tumorous. Okay, and these cells are undergo division to form double the tumor cells. Okay, if tumor cells multiplied, what happened? Complete organ becomes tumor, cancer. Okay, to prevent this cell division of the tumor cells, this drug is usually used. It is like a chemotherapy. Chemotherapy for cancer. Got it?
Yes, ma'am.
Yes. Okay, so this is about the microtubules. So one topic is completed in this cytoskeleton. Second one is microfilament. So microfilaments are solid rods about 7 nanometer in diameter. So it builds a twisted double chain of actin subunits. So these are L4 intracellular movement similar to the microtubule. Okay. And these are L4 long distance transport of vesicles and organelles. Okay. And these are present in intestinal cells. You know, intestinal cells have finger-like projections called microvilli. These microvilli is made up of the microfilaments. But these are the general functions of microfilaments. Okay. So let's discuss the main functions of actin filaments. So actin filaments provide mechanical strength to the cell. Okay. So here there is a cell membrane. A cell next to the cell membrane there is a layer around the cell membrane. Layer next to the cell membrane. So this is the actin filament. So this actin filaments gives mechanical strength to the cells. Okay, and these filaments link transmembrane proteins to the cytoplasmic protein. Ma'am, what it means transmembrane protein, cytoplasmic protein? Cell membrane containing proteins, okay, and the cytoplasm containing proteins. Both are linked. Okay, both are linked to through this actin filaments. Okay, and for example, mitochondria containing transmembrane protein linked to the cytoplasmic protein. That is also through actin filament. Okay.
Yes,
Next, so these actin filaments form contractile ring during the cell division. See, one cell gets separated to form two cells. This separated bridge is called contractile ring.
Projectile ring.
Okay, this is also called cleavage furrow. Got it? Next, these microfilaments L4, cytoplasmic streaming.
Cytoplasmic what?
Cytoplasmic streaming. It is also called protoplasmic streaming. Okay. What it means cytoplasmic streaming means, so transport of cytoplasm within the cell. So one part to other part of the cell, cytoplasm is transported. Okay, with the help of cytoskeletal only, cytoplasm is a fluid matrix, right? It can transport from one place to other place in the same cell. So that is called cytoplasmic streaming. Got it?
Yes, ma'am.
Yes. And it generates locomotion in cells such as white blood cells, amoeba. You know, white blood cell, amoeba undergo irregular movement, right? And they have irregular shapes also because of this actin filament. Please remember, see, all these are very important topics. Okay.
Yes, ma'am.
Next, my actin filaments interact with the myosin, that L4 muscle contraction. This mechanism happened in our body also. Okay, so we are very strong. We are moving. hand is moving, leg is moving because of muscle contraction. The muscle contraction takes place by actin and myosin filaments. Actin and myosin proteins both are very important for muscle movement.
Contraction, relaxation of muscles.
Okay.
Yes, ma'am.
So for actin filaments, the only motor protein is the myosin family proteins. Okay, so myosin has globular head, okay, and tail. So it has the ability to bind to the actin. Look at the image. So this is the globular head of the myosin. Okay, so this globular head have the ability to bind to the actin subunits. Okay, so now, so this myosin helps to transfer substances from one part to other part. Got it?
Got it.
Yes. Okay. So for microtubules, motor proteins are kinesin, dynein. For microfilament, only one, that is myosin, right? And for intermediate filament, there is no motor protein. Then, ma'am, what's the function of intermediate? So
Ma'am, but the
intermediate filament,
intermediate has a lot of proteins, right? So
there
it,
are lot of protein, but there is intermediate filaments do not involved in the intercellular motility. Look at this. Okay, it just provide mechanical support to the entire cell. Okay, it do not contribute to the movement of the cell. It do not transport substances from one part to other part of the cell. Okay, so the intermediate filaments are completely stable, right?
yes, ma'am.
Yes,
And
ma'am.
they are rigid in nature. The intermediate filaments are stronger than the other proteins. Okay, so these are present in all animals, but not in the plants and fungi. You should remember this is also. This is very important point. Intermediate filaments found in all animals, but not in plants and fungi. Got it?
Got it, ma'am.
So intermediate filaments have helical rod-like structures assembled to form the rope-like filaments. Okay. Example, plectin. Okay, so plectin is a intermediate filament. Okay, so this plectin helps to bind the intermediate filament to the other filament. Okay, for example, I told you know intermediate filament and microtubules are associated. Okay, so here this is the intermediate filament. Okay, and here microtubules are there, yellow color one. So these two are joined by the plectin. Joined by a connective proteins are called plectins. Understood?
Yes, ma'am.
Yes. Here connective links are there, no? Say orange thread-like structures. Okay. So these are plectins. So like that, intermediate filaments and microtubules are interlinked. Through which protein? Plectin.
Plectin.
Got it?
Got it, ma'am.
So intermediate filaments, example keratin, neurofilaments in the neurons. Okay, we maintain containing filaments in fibroblast cells, glial cells, muscle cells, and nuclear lamina in the all nucleated cells. Nuclear lamina I explained in the beginning, right? So inside the nuclear membrane, so the nuclear lamina is present. Thin transparent nuclear lamina is present. Okay, so this nuclear lamina made up of intermediate filaments. Clear everyone?
Clear, ma'am.
Any doubts?
Not as of now.
So remember, same with the slides or through the any standard textbooks. Okay.
Mm-hmm.
So
Yes,
then
ma'am.
you will get the clarity in this topic. So different intermediate filament proteins are there, and their distribution given here. See, nuclear lamins, lamin A, lamin B, lamin C. See, different proteins are there. So these are found in the nucleus. Okay. And keratineuronils, okay, hairs, these are contain keratin protein. Right. So that keratin is an intermediate filament. It is present in epithelial tissues. Got it?
Got it, ma'am.
Yes, ma'am.
So now we'll discuss the structural arrangement of intermediate filaments. So, okay, first there is a linear polypeptide site. There is a one protein. One protein is linear in structure. So that is monomer. One end is N terminal, other end is C terminal. So this is a one protein linear structure. Okay. Two proteins joined together. That is called dimer. Okay. Four proteins joined together to form as one bundle. Got it? That is tetramer. Many tetramers to form few bundles. Many tetramers form what? Few bundles. Okay. See, similar to this, many bundles are joined together. Many bundles are joined together like this to form the intermediate filament. So many proteins and many bundles joined together to form intermediate filament. So this is the structural arrangement of intermediate filaments. Okay, read this slide once.
Red, ma'am.
Okay. Any doubt here?
No, ma'am.
No.
Okay. Snigdha, any doubts?
No, ma'am. No doubts.
Okay. So your examples here, intermediate filaments, nuclear lamins, keratin, neurofilament. Read this slide.
Right, ma'am.
So nuclear lamis, okay, it's from the meshwork that stabilizes the inner membrane of the nuclear envelope. Inner membrane of the nuclear membrane. So that gives the specific stability to the nucleus. Okay, so for example, so if there is no nuclear lamis, means what happened? So inside the cell, so nucleus undergo irregular shape. Okay, so that's why so nuclear lamis gives spherical structure to the nucleus.
Ma'am, and also like, does it have to do anything with the ionic concentration and stuff like that? Hypotonic, hypertonic, tonic,
No,
or
no, no. So
no,
that
right?
is due to the osmosis.
Hmm.
Okay, water level in the cell that leads to those hypertonic, so hypertonic and hypotonic. That is due to the water level in the cell. Especially, those that takes place in the plant cells, right?
Okay.
Compared to animal cell, in plant cells, those are very much familiar because of water level in the cells. Plants have the vacuole. Vacuole have the ability to store the water.
Water.
Okay, but animal cells don't have that vacuole. Okay, so that's why hypertonic, hypertonic solutions are common in the plant, rare in the animals.
Okay.
Okay, so not related to these cytoskeleton.
Okay, ma'am.
Okay. Next, the keratin is present in the junction in epithelium. Means two cells joined. These two cells joined through a desmosome. Okay, two cells joined through a protein layer that is called
That's
desmosome.
awesome.
And it also forms a nails, etc. Then neurofilaments. So now cells have long axons. Okay, so these axons are supported by the neurofilaments. That is intermediate filament. Got it?
Got it, ma'am.
So this one slide is enough for the entire today's class. That is
Summary.
read carefully. If you have this slide is enough for revision. If you want, add some extra points to this slide.
Ma'am, I had a question.
Tell me.
Like in the property proteins subunits in intermediate filaments, it is written that one of several different proteins of the keratin family. So can we say that the nuclear lamina and neurofilaments are also
Okay.
part
Also part of the intermediate filament.
are also part of the keratin family because it
Yeah,
is
yeah, yes.
okay. Hmm.
Ma'am,
Yes.
Can you explain one more time what cytoplasmic streaming is?
Cytoplasmic streaming you want?
Yes, ma'am. One more time.
Okay, cytoplasmic streaming nothing but, so in a cell,
Yes.
so there is a fluid matrix that is cytoplasm.
Mm-hmm.
So in that cytoplasm, all organelles are situated, right?
Yes, ma'am.
So your cytoplasm move from, so this part, this place to here, and then transfer from here to here, then transfer to the center, then
Mm-hmm.
transfer to in this direction. In different direction, cytoplasm is undergo movement.
Mm-hmm.
Cytoplasm is undergo transfer. It
Oh.
transfer from one, like move from one place to other place.
Okay.
Okay. So this movement of cytoplasm within the cell, that is called
Okay.
cytoplasmic streaming.
Okay, it's clear. Thank you, ma'am.
Got it?
Yes, ma'am. Clear.
Yes.
Yes, ma'am, understood. Read it.
Yes. Completed.
Yes,
Yes,
ma'am.
ma'am. Yes, ma'am.
Any doubt in that?
No, ma'am.
As of now, no, ma'am.
Okay, that's enough, man. So here some more information is there. Okay. So of the cytoskeletal undergo assembly and disassembly. Okay. For example, see, so each subunit have, for example, actin. Each actin protein have two ends, pointed end and barbed end.
Hello.
Okay.
Yes, ma'am.
And see three actin protein always
Ma'am, I
associate
can't hear
together.
you.
Here what happened?
Hello.
So with the help of energy, yes. This is extra information, not for your exam.
Okay, ma'am.
Okay.
Hmm.
For exam point of this. How the all subunits are joined together. How the protein subunits join together. That I am explaining here.
Mm-hmm.
So here barbed end have the ability to bind with the pointed end. So like that, two subunits joined. Next,
Hmm.
so here pointed end joins the second protein barbed end. So like that, what happened? So here continuous joining, and here what happened? Some the disassembly of the subunits takes place. Okay.
Hmm.
It loss of actin filament joining of the subunits takes place. Here net gain of the filament takes place. Net gain of proteins in a filament takes place. Okay, so like that. So due to the non-covalent interaction, so these subunits are undergo assembly and disassembly. Got it?
Hmm.
Understood?
Yes, ma'am.
Okay, so that's it. So this is about the cytoskeleton. So refer only this topic. Okay, no need to worry
Okay.
about the extra information on this. This is
Hmm.
enough for your exam.
Mm-hmm.
Okay, ma'am.
Got it?
Yes, ma'am.
Okay, that's it. So tomorrow we'll continue with the organization of genes and chromosomes.
Yes, ma'am.
Tomorrow's
Okay.
topic is organization of genes and chromosomes.
Mm-hmm.
Okay, ma'am.
So try to refer the today's topic with textbook. If you have any doubt in that, ask me in the next class.
Okay, ma'am.
Okay.
Mm-hmm.
Bye everyone.
Bye,
Ma'am,
ma'am. Thank you, ma'am.
ma'am, 1 second, ma'am. I have a question.
Welcome. Bye.
Hello, ma'am.
Snigdha, did you receive the study materials?
No, ma'am, I haven't. They haven't added me on the website yet, ma'am.
Just a minute. Just a minute. Just a minute. I resend the materials link. Please
Okay,
join through that link.
okay, ma'am. On WhatsApp? On WhatsApp, ma'am?
Unit 2. Snigdha, unit 2 means cell biology materials link I shared. So in
Okay,
that Google Drive link, all materials are available. Okay.
okay. So
Okay,
should
that's it.
I be
Any
also
other
add
doubts?
GATE website, ma'am? The GATE IIT?
So you want to join to the website portal
Ma'am,
link?
they have not joined me in the website.
Ask to this Jishnu sir. Jishnu sir will guide you. Okay, Jishnu sir will add you in this group. Okay, in portal.
Okay, I don't know how to contact Jishnu, sir. I don't have his WhatsApp number or his contact, ma'am.
Snigdha, I can't hear you.
Hello, can you hear me?
I can, but your voice is breaking. Repeat
Yes,
again.
ma'am. I don't have Jishnu sir's WhatsApp number or contact. Where can I find his
So you
contact?
want Jishnu sir number?
Yes, ma'am.
In WhatsApp, drop a message me. Okay, we'll share you his number.
Okay, ma'am.
Okay, we'll share you his number.
Okay, ma'am.
In WhatsApp, so in this group, I have you know my number. Just one
Yes,
message.
ma'am. I
Okay,
have your
so
number in WhatsApp. Yes.
just to send one message to that number, then we'll share you the phone number. Okay.
Okay, so I'll just message in the group or will message you for Jishnu sir's number? Is
Ah, okay.
it in the group?
Okay. Okay.
Is
Bye, everyone.
there in the group, ma'am?
Yes, yes. Just drop a message me separately.
Okay, 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 done.
Message me by personal number. So I will share you this to sir number.
Okay, ma'am, done. I'll message you.
Okay.
Okay, thank you, ma'am. Bye.
Bye.
Thank you, ma'am. Bye, ma'am.